Okay, so all of you should have received my email with the presentation, what do you call it, evaluation sheet. So I'd like you to pull it up on your computer and make sure you write down the names of the presenters at the top. Today we have Grace and Nigel presenting, who's going to be Grace or Nigel? Who's going to be starting? Who's going to be the first one? Okay, great. So put under presenter 1 Grace and presenter 2 Nigel, and also in the lower part of the form where it says evaluate each presenter individually. For the following, also write both their names. And make sure, not write, type. So I sent you a Word document so you can type in. And at the end, please email it to me. So I take your evaluation as part of the grade. So here, as stated in the syllabus, so the grade will be 50% weight will be my evaluation, 25% weight will be everybody's evaluation, and then 25% weight will be for the presenter own evaluation. So the presenter is going to evaluate themselves. So they get the grade that they give themselves have 25% weight of the final grade. Okay, so that's why it's important that all of you fill out this evaluation. Peer evaluation is important. So we need to learn that, be critical in your evaluation. Also, questions. They are going to be graded on how they answered the questions. And we need to provide enough questions for each presenter to answer. And also asking question is part of your grade as well for your participation. So you get 2 points, participation points for asking questions. So I'd like you to be involved and think about the material that is being presented and how it relates to the theory we learned in class. So today, both Grace and Nigel are going to be presenting under the protein isolation and fractionation category, focusing on one type of protein and looking at the effect of isolation, fractionation conditions on structure and functionality. So this is their topic. So they will start, and Grace and Nigel, you have a maximum 20 minutes, so 16 to 20 minutes. So that's, you're going to be graded on that time adherence. And then we'll have about 10 minutes of questions, and then we can start with today's lecture. Okay, so Grace, anytime you're ready, you can start sharing. And go.
Okay. Okay, well, good afternoon, everyone. I'm Grace Dewi, and my partner is Nigel Kang, and our presentation is on protein extraction from quinoa. Quinoa is a pseudo-cereal native to the Andean region of South America, and it was declared the international crop of 2013 by the United Nations. They are known for their high protein content and are rich in essential amino acids, which made NASA interested in their potential use in deep space missions. So there are two major proteins in quinoa, namely the 11S globulin quinapodin, which is a 320 kilodalton hexamer consisting of acidic and basic subunits that are covalently linked by disulfide bonds. The other major protein is the 2S albumin, and the isoelectric point of quinoa protein is at around pH 5. So as we learn in class, when the pH is below the isoelectric point, most of the functional groups are protonated, so the protein has a net positive charge. Oppositely, if the pH is greater than the pi, then most of the groups would be deprotonated, and that would give it a negative charge. Therefore, at pH above 5, the quinoa protein should be negatively charged. The number of deprotonated groups increases the further the pH is from the isoelectric point. And so the proteins may denature and polymerize at extreme conditions. So pH is an important factor to consider for extraction of proteins.
And yes, like what Grace said, we are studying a little bit more on the effect of extraction pH on quinoa proteins. So we have this slide with our two articles that we selected, and I promise you it's not to confuse you. It helps us illustrate the narrative a little bit more. So we are studying extraction pH, and for these two papers, the first paper focuses on effect of extraction pH on heat-induced aggregation, gelation, and microstructure of protein isolate from quinoa. And they use a acid precipitation method as a way to extract the proteins. And for their objective, they were trying to link their findings to food applications. And in the second paper, it's the same thing, revolving around pH, except they have this treatment, which is the ultrafiltration treatment, which is in contrast to acid precipitation. So separation by size instead of separation by the isoelectric point. And these two papers have pretty similar sample preparations. They started with quinoa seeds. The only difference is that the second manuscript has to remove the saponins that are inherently present in the quinoa seeds, and these saponins can interfere with the functionality and the findings. So they had to co-wash it and dry it to form the sweet quinoa seeds. And from then on, both papers prepped the sample similarly. They ground the seeds, they sifted them, and produced the flour. And obviously after that, they had to defat the flour to prevent interference with their assays and other processing steps. And then this next slide over here is just like a combination of both the two papers and what they did. I've separated what they did to help visualize how they performed their experiment by color coding them. So the green is for the first paper and the orange reflects the second paper. So they started with the defatted quinoa flour. They extracted them at pH ranges of a pH range of 8 to 11 to solubilize the proteins. And then they centrifuged them, collected the supernatant. And then from here, the two papers differed in the way that they extracted or actually separated the proteins. So for the first paper, they did acid precipitation to get the isolate. And then, lowering down to the isoelectric point where the proteins start to lose their charge and then aggregate by hydrophobic interaction. And then the second paper, they did ultrafiltration. So what they did was either maintain the pH at 9.5 or lower it down to 7, and then subject it to the ultrafiltration steps to get the different fractions, and then freeze drying them to get the extracts. So over here, the flow chart on the left is basically what I mentioned before. I'm going to be explaining about the first paper. So this is for the first paper. They quantified the protein using the Dumas method, and then they also characterized the proteins by molecular weight, measuring the thermal properties, observing the microstructure, and also performing gelation tests. So for their first result, they measured protein by a crude method, the Dumas method. So as we can expect with increasing extraction pH, we see more yield, and that is due to the higher net charge of the proteins, allowing them to get electrostatically repulsed from each other and then having better solubility. But interestingly, we see a decrease in the purity, and the authors attributed that due to co-precipitation of different substances out of the solution. And then they measured the size distribution of their isolates. What I want to point out here is that at 50 kDa, we see the globulin bands. And then Also at 200 kDa, we see these bands that were not able to travel down because they're too heavy. And then comparing the 4 different extraction pH to the defatted quinoa flour, we see that there is an obvious structural difference, just looking at the intensity of the band and also just the locations of the band. And one thing to note is that these proteins that were loaded into the gel were not corrected, so they didn't have equal protein concentration. So it's also very hard to make conclusions because it's not a fair treatment. But based on this result, we see that there is hydrolysis that occurred as we see the decreasing protein intensities, the band intensities as pH increase. So for E8, we see a higher distribution of higher molecular weight proteins, and for E11, we see a lower distribution of high molecular weight proteins. And the authors also noted that polymerization was not observed here because when proteins are subjected to extreme pH, we see polymerization due to extensive denaturation that causes the exposure of hydrophobic groups and sulfhydryl groups that cause them to aggregate. So in the step where they centrifuged them, it got separated, so it didn't get into the supernatant. And then for the thermal properties, they measured using DSC. So as we learned in class, with increasing denaturation, we see a smaller heat absorbed, which is the change in enthalpy over here. They all had the same denaturation temperature, as these all resemble the globulins. And for E11, we don't see a peak because it has completely denatured essentially. So we don't see any heat absorbed to cause disorder in the chain. And then this chart on the right here is just a different way of expressing the results on the left. And then for solubility curves, below the PI, we see that E8 is the most soluble, but above the PI, we see E9 is the most soluble. But overall, there's not many major differences. And the reason why we don't see a lot of solubility for E10 and E11 is due to the same reasons we don't see a lot of intensity for the extreme pH bands in the SDS-PAGE. So the authors linked it to those results, saying that there was aggregation, so it wasn't present. It wasn't able to solubilize. And also they use a 1% weight by weight protein solution. And then for the gelation test that they did, they measured the particle size. For E8 and E9, we see an increase in the particle size, and that's indicative of association and aggregation, also indicating that there's a formation of a gel network, which is also reflected in the right graph over here. We see how they did gelation. The temperature curve over here tells you what temperature that they use. So they heated it to denature the proteins and then cooled them to form the gels. And they kind of linked the results here. They see that there's a strong gel formation for E8 and E9. They kind of linked it to the DSC results. So in the DSC results, they saw that the denaturation onset was at around 75. And then for this result over here, we see a gelation onset at around 70 degrees. So they kind of linked it there, indicating that denaturation occurred before the formation of the gel, because this is a heat-induced gel. And then they also observed the microstructure. The green represents the protein phase, whereas the black represents the background or the water. We see a trend for the lower pHs. They see a strong gel formation, supporting the other results. We see big agglomerated particles that form a nice network. And then for E10 and 11, they found a finer particulate size distribution, which attributed to, which caused a weaker gel. So over here, the table on the left is just a summary of what I said before. And the authors ended their paper concluding that E8 and E9 have possible semi-solid gel food applications, whereas the higher extraction pHs are more applicable in liquid foods. But my critique to that, especially for their findings on E10 and 11, the lower solubility curves didn't seem to support that conclusion, because for E10 and 11, we see a lower solubility there. And then the concentrations that they use, which was 1% protein solution, is not applicable in foods. It doesn't reflect reality. And for the SDS-PAGE, they used unequal protein concentrations, which didn't make for a fair treatment and a fair comparison of the protein bands. So what we had out of this was we needed further studies to explore the effects of pH on protein purification. And since they concluded on the types of food applications that they could have, so we were wondering on the scaling up of quinoa protein extraction. And that brings us to the next paper.
Yeah, so the second manuscript by Navarro Lisboa et al. Investigated the ultrafiltration technique using a ceramic membrane with a 300 kilodalton molecular weight cutoff. So filtration was tested at either pH 7 or 9.5, and the three fractions that were analyzed were the initial pH-adjusted sample, the concentrate, and the permeate. The fractions were analyzed for permeate flux, protein content by Bradford, molecular weight composition by SDS-PAGE, thermal properties using DSC, and Zeta-Potential for surface charge and interfacial tension. Other analyses will not be discussed for the sake of time. But coming to the first result, so this graph illustrates the permeate flux for the two different pH. Here, the steeper curve observed at pH 9.5 showed that the permeate moves faster through the membrane at pH 9.5 than at pH 7. And the authors concluded that this may be due to the closer proximity of pH 7 to isoelectric point compared to 9.5. I included a graph representation for all the results that were presented in table form to help visualize the differences. So these graphs will have pH 7 in green and 9.5 in blue, and along the x-axis are the different fractions that were quantified or analyzed. And as expected, the highest protein concentration was observed in the concentrate, and more proteins were detected at pH 9.5. So looking at the Zeta-Potential, proteins had a greater net negative charge at 9.5, which is expected since it's further from the isoelectric point, and thus more groups would be deprotonated. The authors also mentioned that this may contribute to the faster movement through the membrane, as less fouling would occur with greater charge. Next, we have the SDS-PAGE, which was conducted under reducing conditions. The first three lanes after the molecular weight marker are the initial, permeate, and concentrate fractions collected at either pH 7 or 9.5. The authors noted that the permeate at 9.5 had bands at about 17 kilodaltons that were not observed at pH 7. There were also larger molecular weight bands between 55 and 34, which were absent in the concentrate at pH 7, and the authors noted that these larger proteins may have been deposited to the membrane. The bands attributed to the acidic and basic subunits of the globulin were observed at the concentrate in both pH. The authors mentioned that the presence of the protein under 300 kilodaltons, which is the cutoff of the ceramic membrane that they used, is due to the selectivity of the membrane. However, they did not discuss the fact that it was only done under reducing conditions. And as Pam noted in class, they should also run the samples under non-reducing conditions so as to not overestimate the smaller molecular weight proteins. Moving on to the thermal properties, these graphs illustrate the denaturation temperature and change in enthalpy results based on the data from table 3. So the denaturation temperature was lower at pH 9.5 compared to pH 7. However, they reported no statistically significant difference in terms of enthalpy change between the two pH, though it was lower in the concentrate fraction. Notably, the denaturation temperature reported is above 100 here, whereas in the paper that Nigel discussed, it was below that. And the authors attributed this to the low water content. However, it's worth noting that they directly analyzed the freeze-dried samples without resuspending them in solution. The following graph illustrates the measured interfacial tension for each sample at the same time point. So here they observed lower interfacial tension at pH 9.5, which they said may indicate a better ability to adsorb at the oil-water interface. They attributed this to the decrease in surface hydrophobicity, but they did not measure that directly. So in summary, they observed that permeate flux, protein concentration, and net charge were greater at pH 9.5. The molecular weight information is inconclusive as they did not analyze the samples at non-reducing conditions, and denaturation temperature and interfacial tension were lower at pH 9.5. With this, Navarro-Lisboa et al. Concluded that ultrafiltration and pH affected quinoa protein extracts, and that the pH was the most important factor to obtain specific characteristics. So one of the caveats of this manuscript was the unclear rationale behind the experimental design and methods. They also barely discussed the ultrafiltration technique with regards to their results, despite it being a key component in their study. Overall, we'd recommend that they design their experiments with their objectives in mind, and it also highlights the importance of understanding the principles behind the methods, especially when it comes to the analysis and presentation of data.
So the overall takeaway that we got from this whole study of the two papers is just the importance of monitoring the structural changes that occur throughout food processing, and of course, since we're taking a food proteins course, also when it comes to experimental design, knowing how those structural changes can impact our functionality. So having a good experimental design that reflects real food systems and makes sense, and knowing what exactly are we measuring and what are we trying to find, and whether it's a good indicator of what we're trying to find is important. And also just the ability to critically scrutinize the author's interpretations in papers, recognizing their assumptions, identifying inconsistencies in their findings, and then also being able to address the elephant in the room. So for example, like earlier when they concluded in my paper, paper 1, that they concluded that the higher pH were more suitable for liquid food applications. They didn't consider the solubility curve. They just went on the basis of the gelation results. So, and then lastly, just learning from other researchers, the good things that they did, the bad things that they did, the pros and cons, and learning how to apply that into our work as graduate students. And with that, I conclude our presentation.
Okay, good. Right at 20 minutes. Okay, good. So we can open it up for questions. So unmute yourself and ask your question.
I have a question. Oh, am I talking over someone? Sorry, I can't
No,
tell.
no, no. You can go ahead, Laura. I can ask.
Okay. Yeah, one of the things I found interesting with the Ruiz paper was how long they solubilized for. I think it was something like 16 hours. Do you guys have any thoughts about that, as like how that might have affected their results or reasonings for solubilizing for so long?
Right. I can go ahead and answer this question. So with the extensive alkalinization time that they did, 16 hours compared to the other paper that did 4 hours. So that was a big difference. They found that there's a lot of denaturation that happened, which was also indicated in the SDS-PAGE. And that also affected the yield when they were comparing the yield to other studies, other quinoa protein studies. So yeah. Any other questions? I think Brigida had, or Sonali.
Yes. Yeah, it was a really good presentation, but I just wanted to ask, what differently would you have done if they had given you the same objective? Like from what you learned in the class, what differently would you have done? Because I did see the critiques that you guys gave. So what differently would you guys have done in either of the papers? It's not particular, so.
So for the second paper, there was probably like a lot of things that we would have done differently. One thing that particularly was kind of like the center point was that the fact that they only ran their SDS-PAGE under reducing conditions, which was a question that was brought up in class. And in almost all the other papers that I've looked at exploring using SDS-PAGE under reducing conditions, they'd always present it with like one that didn't have any reducing agent added. And based on the discussion from the second paper, it kind of came across that like I don't think the authors realized that they did it in a under reducing condition because they never mentioned that. They mention other people's experiments, but they don't reflect on their own. So that, and the other thing we were also questioning for the second paper is why were they interested in the permeate if like the main point of ultrafiltration is to get the concentrate. So those are some of the two.
Yeah, I have the same points as well. Just maybe being more critical about the way that they're approaching their experiments. For SDS-PAGE, they had to use equal protein concentrations, obviously. And then for solubility, using a more realistic concentration that is not just 1%.
Come on, you guys. I'm going to take -2 of you if you don't ask questions. I need more participation. This is crucial to show that you've been listening. You are going to critique the work and you're going to ask questions. I'm going to hold you accountable because it's important. It's part of the whole experience.
Grace.
So Nigel, sorry.
Go ahead.
Okay. So Nigel, I saw that your paper had less functional methods since your paper also wanted to discuss as one of its objectives is finding applications for it. Do you think they should have run more functional assays, or what more would you have wanted to run with that?
Yeah, definitely. I wish that they perform some sort of like Zeta-Potential maybe, because knowing the charges on the surface charge is important for gelation. I think that would be a good way for them to link their findings on gelation as well. And yeah, like maybe possibly even since they were talking about beverage applications, probably looking into emulsifying capacity, emulsifying ability and stuff like that. Yeah.
I just want to say something. Zeta-Potential is not a functional property. It is more structural, physical-chemical property that is basically telling you the charge of the protein, the net charge at a particular pH. So yeah, it's more structural, physical-chemical characteristic than functional. Okay, Kenzie, you had something to ask?
Yeah, Grace, in the part that you were presenting about, you mentioned a couple times what they said about like the membrane flux at pH 9.5, and I kind of like missed it a bit. So I was wondering if you could tell us again why they thought that.
Oh, like why the proteins, why the permeate flux was greater at pH 9.5?
Yes.
Yeah, so their hypothesis is that because at the higher pH, the proteins have more negative charges on them, the repulsion forces will kind of help make the make the proteins move faster through the pores. And they also mentioned that there would be less polarization, which is basically when the proteins kind of clog up the pores. So that's why they're thinking that at pH 7, when it's a little bit closer to the isoelectric point and maybe a little bit less soluble, then the proteins at that pH would kind of like clog up the pores, and then that's why the permeate flows a little bit slowly, and you see this kind of like a flatter curve with pH 7, and it also took longer for them to get to the same volume concentration factor. Pam, there's a question in the chat. Is it okay if like?
Yeah, go ahead.
Okay.
Yeah.
So Chrissy asks, how did you decide on which papers to discuss? And And were there limited articles on quinoa? Were you interested in quinoa in particular? That's a great question. We were contemplating multiple types of protein, and quinoa was one of them, that one of the top ones that we were interested in. But then we, after like once we selected the papers and try to look, well, we found one paper that we really liked, and we tried to find a complementing article to it. And then that's when we kind of realized that quinoa was big around 2013 and kind of like a lot earlier. So some of the better papers that we wanted to pick were a little too old for the class project. But yeah, so we try to make do.
Yeah, I know. It's just you select a paper, you read through it a little bit, and it looks good. But once you dive into the results, we find out that, oh, it's not as good as we thought. So you called, I have a question. So you called it ultrafiltration, but the pore size is 300 kilodalton. This is nowhere close to ultrafiltration. And how come the proteins did not go? I mean, some proteins went through, but how can you explain that a lot of the protein were in the retentate when you have that large of a molecular?
So I think I can answer this question. What Grace was trying to say as well earlier, I just want to add that the ceramic membranes, they theorize that when the proteins are like clogging up, the proteins were clogging up the ceramic membrane because once they kind of like absorb onto the membrane itself, they kind of unfold, and then they expose the hydrophobic groups, and that causes more aggregation as the proteins are flowing through, and that created like an aggregate, and that clogged up the ceramic membrane. So we saw more of the retentate in there.
That's interesting because they don't show polymers in their gel on the pH 9.5. So where did these polymers go?
I guess would that be partially because of the mercaptoethanol that they added for running the SDS-PAGE? So then you wouldn't see any of the larger ones because it'd be like the disulfide ones would be broken. And
yeah, and hydrophobic interactions will be broken too with the SDS.
Oh, and with regards to your the question about why they called it ultrafiltration, I think that's quite like indicative of how the authors, I don't think they have much of a food science background. So some of their methods, I think they had good intentions, but they're not fully like clear on the principles. So that affected how they interpreted their results and designed their whole experiments.
Yeah. Yeah, I mean, I think their results are questionable, especially when they do measurement at not the same protein concentration. So when we run a gel, in order to compare, we always run it at equal protein concentration, and neither of the two papers did that. So it's something to note for everybody, is like this is a major thing that they didn't do. Yeah. Any other questions from the group? We have maybe 1 minute.
Yeah, just maybe it's a dumb question, but under the SDS-PAGE results, let's say on the, let's pick my book. The paper by Navarro Lisboa, about third paragraph down, it says that the proteins below the membrane cutoff remain to concentrate, where the proteins smaller than the cutoff pass through into the permeate. That just seems like it's worded kind of funny.
Yeah.
I didn't understand it, what they're trying to get at there.
It's an error.
Yeah, and actually on this one too, acidic and basic subunits are highlighted in red because in their discussion, they actually switched the two and they referred to them incorrectly. So I think they were a little bit confused.
Yeah, there's a question from Radhika. Are ceramic membranes commonly used for protein fractionation?
Great question. I wasn't able to look too deeply into that, and I'm not sure. They also didn't really discuss the use of ceramic membranes besides kind of like their benefits, which part of it is because of its versatility, that it can accommodate like a wide range of pH and a wide range of temperatures. But beyond that, they didn't really explain why it had to be ceramic.
Holly,
have a-
do you have a question?
Yeah, can you go to the slide on protein content for manuscript 2? Okay. Is this like, do you have, did this paper give purity for this method?
No.
Okay. Do you have any insight as to what the purity of this method would be versus traditional like pH alkaline acid precipitation extraction?
Great.
Just because,
Oh.
yeah, no, I was just gonna add and say like, because if you're only solubilizing at a certain pH and then your ultrafiltration is your purity step, but your membrane is 300 kilodaltons, like, would you expect to see anything other than just protein in what you're collecting?
That's a great question. I'm not sure. Nigel, do you want to chime in?
Well, I think that there would be impurities in it, like physically, like they would be present there because of all the fouling that's happening. I'm sure that there will be other components that would get caught in the membrane. So yeah,
Didn't
I
they
would.
solubilize first? Didn't they solubilize the protein first and then ultrafiltered? Or filtered, I'm not gonna call it ultrafiltered. Didn't they solubilize the protein? At what pH did they solubilize the protein?
Yeah, so they extracted at 9.5 and then they adjusted the pH after they collected the supernatant. How they adjusted, they didn't explain.
Yeah.
So maybe like there would be co-precipitation of like other substances when they extracted it at 9.5, maybe. Could be possible.
I mean, there
Yeah.
would be some other components that would be soluble, maybe some soluble fiber, soluble sugars, there might be some, but smaller molecular weights will potentially go through that ceramic membrane. Yeah, they didn't look for purity, and that's something that they should have done, basically. Yeah. So Grace, there's a last question, and then we'll stop here. That's from Devin. Your paper seemed more interested in testing the filtering membrane. Is this the hole in the literature that they defined in the intro? Did they relate the change in functional properties to its application at all?
Yeah, so that was kind of the purpose of the paper, was to investigate the filtering, like kind of, as Nigel said, as a way to scale up the extraction process. And that was the entire premise why we selected this paper, because we were interested in this other side of the extraction process. And I think at some point they kind of lost sight of their objective, because at the end, they're really more discussing the pH than the filtration process. And they didn't really relate much of their findings with regards to the membrane. And if they did, they didn't measure it directly. So it's kind of like assumptions and comparisons to other applications from other papers that they themselves did not investigate. So
yeah. They did only interfacial tension as the only sole functional property. Okay, so we don't like that second paper, but it's okay. You did a good job trying to explain the results better than they did. So it's okay. We learn just as much from not that great papers as much as from a good paper. Okay, thank you. I guess I'm just gonna give you guys 5-minute breaks now, and then we'll do about 60 minutes of lecture. So we'll come back at say 3:40. That's seven minutes.
I do have a question on the paper. Can I ask you?
Yeah,
Okay.
sure.
So on the extraction method, I realized that they ultrafiltered at different pH. And then I was wondering if like before freeze drying, did they adjust the 9.5 treatment to like 7 pH?
They
pH 7?
didn't. They
So
didn't. And I don't think they did freeze drying either, did they?
they did in the paper, I think.
Yeah, they freeze dried the samples.
Yeah. So they
Oh,
freeze dried at 9.5 pH. Is that a good thing?
no. That's not a good thing. You never want to freeze dry at alkaline or at acidic pH. So we always tend to bring it close to neutral before you freeze dry.
I was
So
wondering if that was why there was a difference with the DSC, like the at 9.5, it denatured at a lower temperature compared to.
well, yes. Well, also they don't look at salt content at all, because if you have more, depending on the salt, ash amount and the salt content, the denaturation temperature changes. So yeah, so they didn't do a lot of testing that might explain their observation. So we're guessing at this point what happened.
Okay. That's weird.
Okay, yeah, well, good. All right, I will 5 minutes and get back to lecture.
Okay.
Okay, let's get back here. So I will share my screen. Okay, so today we're starting a new topic. And this is our map for the class. Like I always show it just to show you where we are so far. So we talked about a protein structure, intrinsic characteristics, effect of external or extrinsic factors on the structure, and we talked about functionality, and we related structure to function. We had a lecture on enzymes because enzymes are mostly food-related enzymes are proteins and also are used on a lot of applications. And now under intrinsic factors that we haven't touched on a lot, I gave you examples throughout, but now we're going to dive into the source of the protein. And we have several sources on the syllabus that we will be hoping to cover. And I'll see how much I can cover between now and end of the semester. So today we'll talk about dairy proteins or milk proteins in more detail in terms of the structure, the different protein components, their structure, their functionality, how they're produced, the ingredients, the dairy ingredient, protein ingredients, and then some applications at the end. Okay, so just as a reminder, I showed you this at the beginning, I think it's first lecture, that we have different protein ingredients in the market, and we'll try to cover as much as we can, as I said. But if we look at dairy proteins in the market, there are sodium caseinates, calcium caseinates, micellar casein. These are the main casein ingredients. And the other big ingredient from dairy is whey protein. And we have whey protein concentrates, and they range in protein purity from 35 to 80. So you call them WPC 35 or WPC 50 or WPC 80. That means how much protein is in that concentrate. And then we have the isolates that have 90% protein purity or higher. In soy, we have three main ingredients. The flour is high in protein, so it's 50% or 50 to 60% protein, especially the defatted soy flour. So it is considered a protein ingredient. And then you have the soy protein concentrate, which is about 60 to 65%, can be a little higher too. And then you have the isolates, soy protein isolates usually are above 90% protein. Then we have the egg protein, the albumin, and then the yolk, cereal protein, vital gluten is used in baking applications. It's used for texturization, for meat analogs. So it is another important protein ingredient. And then we have the meat-based proteins, gelatin, myofibril proteins as well. All of these, or most of them, you would find hydrolysates. So you find whey protein hydrolysate in the market, you find soy protein hydrolysates, you find albumin protein hydrolysate, and even gluten protein hydrolysate. So it is another form of an ingredient for enhanced functionality. So new and emerging sources, there are a lot right now, and big ones are pulses, and peas is one of the main ones. So I will potentially, when I cover legumes and oilseeds, we'll talk mostly about soy, but I will bring in some information mostly about pea because it's gaining a lot of traction. Oil seeds, we'll talk about soy, we'll touch base on some of the other oil seeds that are emerging. Of course, there are others that we won't get to, but there are a lot of different protein alternatives to the ones that are traditional right now, but we won't get a chance to talk about all of them. When we talk about grain proteins, I might talk about wheat, might bring in some oats, but we won't definitely be able to cover everything that is out there and might become an important ingredient. Okay, so milk proteins. So in milk, there are so many different types of proteins. It's just a large number of proteins. However, the main components that are functional proteins and we care about and we produce ingredients from are the casein and the whey. And the casein represent 80% of the total protein in milk. So you don't have really high percentage of protein when you talk about liquid milk. It's mostly water, right? 87% water. But when you look at the protein component, 80% of that is casein mostly, and whey represent the 20%. The other minor proteins, they're too small an amount, too little an amount to contribute in a significant percentage. And there are a lot of enzymes. And they present a small concentration, but they are impactful enzymes. There are a whole list of enzymes present in milk: oxidoreductases, transferases, we have hydrolases. I circled here proteinases, especially plasmin. This enzyme has consequences on the quality of milk. It impacts shelf life of milk. Over time, plasmin hydrolyzes casein, especially beta casein, causes thinning of the milk and gelation. So sometimes you see separation of the milk, and that is due to the action of plasmin on the caseins. Also, plasmin is important in cheese ripening. So it does proteolysis during cheese ripening that will also impact the flavor. So it's a very important endogenous enzyme in milk. Another one I highlight, of course, there's lipase, there is lysozyme, but the one that I circle is alkaline phosphatase, and it's an important enzyme from a processing perspective, because when you pasteurize the milk, you're aiming at killing pathogenic bacteria. And instead of doing a microbial test to determine the efficacy of pasteurization, you can look at the activity of alkaline phosphatase, which is more stable than pathogenic bacteria. So if it's inactivated, that's an indication that pathogenic bacteria has been killed. Okay, so that's why it's important, and I circled because it's an important enzyme that is used to test the efficacy of pasteurization, milk pasteurization. So a lot of enzymes. If we want to, we'll dive more now into the major proteins that are important for functionality, and then the different ingredients from that are milk proteins ingredients. Okay, so casein proteins mostly are 4 different types of caseins, and they're alpha S1, alpha S2 casein, beta casein, and kappa casein. So we'll learn about the structural differences of these different proteins and how each impact the functionality. So what I have here is a gel, and this is a urea PAGE gel. Why I put it here is I just wanted to show you the different caseins. So you have the A, B, and C here. So alpha casein right here. So they don't separate very well, alpha S1 and alpha S2. And then the B here is beta casein, and then here is kappa casein. So normally when we want to look at caseins proteins, we don't do SDS-PAGE because SDS is basically separation mostly based on size. And these proteins are all very close in molecular weight. So ranging between 19 to 20, 21, 22 kilodalton. So they are really, you cannot see, you see as one blurb or one big huge band on SDS-PAGE. On urea gel, separation is based on size to a charge ratio. So they are different in their net negative charge. We'll see that in a little bit when I talk about each one of them. So we can separate the alpha from the beta from the kappa, kappa casein, but not so much the alpha S1 and alpha S2. We don't see that much of a separation. This lane here is sodium caseinate isolate. So the sodium caseinate isolate, which we'll talk about how it's produced later on, it mostly has beta and alpha casein. There might be some kappa casein, but it's probably in very low concentration and it doesn't show that well. Okay, so that is the different types of caseins. Now, whey protein, there's so many different whey proteins, but that means the protein, they call them whey protein because these are the more soluble proteins. So when you form a curd either by acid precipitation of casein or by enzyme formation of cheese by chymosin, you get the whey, the water portion that has proteins, has lactose, has some minerals. So they call them whey proteins because they're the soluble proteins in whey. So mostly, most of the whey proteins are beta-lactoglobulin and alpha-lactalbumin. So here at the top gel that I show you, the first lane here is what I want you to focus on. Don't worry about 3, 4, and 5, just 2, lane 2. So here the beta casein is beta lactoglobulin is E, and the alpha lactalbumin is F here, that is labeled F. So the beta is anywhere between 16 to 18 kilodalton in molecular weight, and the alpha is between 12 to 14 kilodalton, depending on the variant, genetic variant. You can see also this is a whey protein concentrate actually, but you see that mostly beta-lactoglobulin is present in higher concentration than alpha-lactalbumin. But also it varies in different isolation, purification of the concentrates and the isolates. For example, if we get whey protein isolate from Agripur, it is isolated by ion exchange. It has more beta-lactoglobulin and alpha-lactalbumin. But in milk per se, mostly you have about probably 60, 70% to 20, 30% beta-lactoglobulin to alpha-lactalbumin. Other proteins are there in whey. There are minor ones, and then there are some that are a little bit present more than the little other proteins, minor proteins. So here in the gel, A here, right there is immunoglobulin, and then lactoferrin is B, and then serum albumin is C. Okay, so you have these proteins and you can actually see them on a gel as well. They're not as prominent, they're not necessarily, they don't have functional properties, they might have biological properties, but the functionality of whey protein isolate or concentrate lies upon the beta-lactoglobulin and alpha-lactalbumin, and that's why we will focus more on that. This is another chart, it's more breakdown of the different proteins in milk. Minor proteins, enzymes, but mostly you have caseins and whey protein. Okay, in caseins, you have the alpha S caseins, alpha S1 and alpha S2. So this A, B, C, and D is basically genetic variants. Kappa casein, you have 2 genetic variants of it. You see here beta casein, you have multiple genetic variants, but then you might say, okay, so what's under beta casein? You have these gamma caseins, and then here you have proteose peptone. Okay, so the gamma caseins and the proteose peptone are breakdown due to endogenous enzymatic hydrolysis of beta casein. You get the proteose peptone is soluble, is hydrophilic. So it stays with the whey protein. If we're separating the casein from whey, it winds up in the whey because it is hydrophilic. The gamma caseins, depending on their variants and where the enzyme cuts, are hydrophobic. So they stay with the casein fraction. And these are generated mostly by the plasmin enzyme, which I talked about. It's the endogenous enzyme. And, you know, during storage of milk, this happens. So you always have some form of hydrolysis that takes place. And you have these different peptides generated. The hydrophobic ones remain with the casein, and the hydrophilic one remains with whey. So under whey, we talked about there is your serum albumin, beta-lactoglobulin, you have different variants. Alpha-lactalbumin, you also have different variants, and then you have immunoglobulins, and you have different types of immunoglobulins. Okay.
So, I have a question.
Yeah.
How are these variants different from each other? Do they just have different sequence of
Yes.
acids? Okay.
Yeah, so there is a lot of homology among them, but there could be some differences in amino acid composition total and also in the sequence. Mm-hmm.
So could variants be analogous to different protein isolates from different cultivars in plants, or are they not the same?
I mean, I don't know if it's analogous, but it's the same concept. Like every cow has certain genetic material. And usually when you get milk from multiple cows, you'll see multiple variants. But then if you take one cow and take the milk from one cow, you'll have one genetic variant usually, unless a cow is unique. But yeah, so cultivars is different. You have environmental, same thing with cows. What are they fed? Where are they grown? And plants the same way. What soil they're grown in, what's the environmental conditions, what's the varietal difference. But I can't really compare. It's not apple to apple comparison in terms of the amount of difference and the significance of the difference. I can't relate, but kind of the same concept. Different varieties of cows will give you different variants of these proteins.
Got it.
Okay. So we'll talk about caseins. All right, so a very important characteristic of casein is phosphorylation. And I mentioned it earlier when I was talking about the reactive groups, serine and threonine. They have this OH groups that can get conjugated by following post-translational modification. So phosphorylation, mostly on the serine in these casein, is very characteristic. It kind of influences the formation of the micelle, because then you have phosphates, and then phosphate interact with calcium, and you have calcium in milk, and then you have the phosphate calcium bridges within a casein micelle that's among the stabilizing forces. So this is a very important characteristic. Beta casein is unique and different than the other caseins in a sense that it is the least phosphorylated, so it's the least charged, and it's the most hydrophobic. I'm going to show you most of its sequence is hydrophobic amino acids. So that is very characteristic of beta casein, and it is the driving force of hydrophobic interactions within the caseins in the micelle. So this is back to phosphorylation. They are all of these caseins are sensitive to calcium, so we can potentially precipitate them by adding more calcium to the milk. So it's kind of like we can destabilize them by having calcium. To connect that is when you're making cheese, you add calcium sometimes to your milk to enhance curd formation. Kappa casein is glycosylated, not phosphorylated. So it has a carbohydrate moiety attached to it on mostly threonine amino acids. So it is very important characteristic as well. And the kappa casein, it's kind of the casein that stabilizes the micelle. And I'll show you that later. I mentioned it earlier. It has the glycomacropeptide, which is the part of the casein that is glycosylated. It's highly charged, and it mostly is on the surface of the micelle, increasing the zeta potential of the micelle, stabilizing the micelle as a colloidal system in milk. So kappa casein plays an important role in the stabilization of the casein micelle, preventing it from interacting with each other. So they stay suspended in milk unless they're hydrolyzed or heat treated or acid treated, then they lose the stability. And we'll talk about all of that. So what other important characteristic about all of these caseinate are their random secondary structure and open tertiary structure. So reminder, they have high percentage of proline. And the high percentage of proline breaks the formation of an ordered secondary structure. And again, you want to remember that your R chain is covalently linked to your amine group, providing rigidity and lower flexibility of the bonding and the turning of the different bonds that allow formation of alpha helix or beta sheet. So it's mostly an open structure, and the structure is highly amphipathic or amphiphilic. They mean the same thing. That means you have clusters of hydrophobic regions. So you have hydrophobic regions, very characterized hydrophobic regions, and then very characterized hydrophilic regions. So regions or clusters, you can call them whatever you want. Excuse me. So what this characteristic makes them what?
Good emulsifier.
Yes. So there are what we call them natural emulsifier. They're open structure, they're flexible. So they have the hydrophobic and the hydrophilic groups, very amphiphilic. So they kind of emulsify naturally. You have high surface adsorption. Okay. So let's dive a little deeper in the structure of these caseins. So if we look at alpha S1, so you have about 7 to 8 phosphate groups, which increases the negative charge on that protein. And I don't necessarily want you to remember the sensitivity, like at what concentration of calcium it kind of precipitate, but it is sensitive to calcium, but not as much as alpha S2. We'll see it has higher phosphorylation. The higher the phosphorylation, the more the sensitivity to calcium. So it has 3 distinct hydrophobic groups. So this white region that doesn't have any lines or is not grayed out is hydrophobic. So hydrophobic amino acid, mostly hydrophobic regions. The region that has these lines, these are negatively charged mostly. So you have a net negative charge of -20. And this region that is grayed out is hydrophilic, but not charged. So this is the characteristic of that one. The alpha S2, of course, this is not how it existed. It's just like I'm explaining it as a strand of polypeptide. So alpha S2 is the most phosphorylated of all caseins. And because of that, it has the highest sensitivity to calcium. So at lower concentration of calcium, you can get it precipitated. It's the most hydrophilic because it's the most phosphorylated, that's one. And it has multiple regions that are negatively charged. So again, these lined regions are negatively charged, and then the grayed out regions are hydrophilic. But it does have the C terminal is highly hydrophobic. So it's still a very amphiphilic protein. So what's unique about alpha S2 compared to alpha S1 and beta casein, it has a disulfide, one disulfide. So it has two cysteine residues that are, they are connected by disulfide linkages in the molecule, but they can take part in molecular interactions. So if we get any free sulfide group close by, they can cause disulfide interchange. So that is unique about alpha S2 that is not in alpha S1 or in beta casein. They don't have cysteine residues. So beta casein is the most hydrophobic. So if you look at the strand of the protein, most of it is a hydrophobic. It's very amphiphilic. So it has a very concentrated region of hydrophobicity, of high hydrophobicity, that is. It's all of this part. And then the N-terminus is charged, negatively charged. So the net charge is -11. So it's the least charged. It is phosphorylated, but it's the least phosphorylated protein. So it's most hydrophobic, least phosphorylated, very amphiphilic or amphipathic, same thing. It has the highest proline content, so the most random structure. And it is the most casein out of all caseins that is susceptible to plasmin hydrolysis. So there are certain specific regions. So plasmin has specificity similar to trypsin at lysine and histidine locations. So it breaks at 3 unique places on the protein. Hence, you have 3 gammas, and you have the hydrophilic protease peptone that end up in the way region or way component. Okay, so kappa casein, it is also very amphiphilic, but instead of being phosphorylated, it's not sensitive to calcium, so it doesn't have phosphorylated groups. It has carbohydrate groups, and its C terminal in this case is highly charged, and it's glycosylated at threonine residues mostly. And these are the different carbohydrates that would be found on the kappa casein. It plays an important role in the stability of the casein micelle in terms of the hydrophobic interactions. It drives the hydrophobic interactions that link those proteins together and keep them stabilized. So you have the kappa casein outside stabilizing with the glycomacropeptide. And inside you have the calcium phosphate bridges between the phosphorylated groups on the protein, and then you have the hydrophobic interaction, which is it's very important force of stabilizing the casein. So this part of the casein is the glycomacropeptide. So you'll hear that term. It will come across. It mostly it winds up in whey after formation of curd with enzymes, because the enzyme is going to cut off at the glycomacropeptide here, and this part, the glycomacropeptide, will end up in whey, and the paracappa casein. This is that. It stays with the curd, with the casein curd. Okay, so this is just a slide that, you know, put them all together and show you the differences. And this number here is the number of amino acids in these different proteins. Okay, so it just tells you, well, the kappa casein has one phosphorylated group. I don't know if I said it's not phosphorylated, but it has one, but mostly it's the carbohydrate glycosylation here at this end of the protein. Okay.
Oh, Pam, just for clarification, on that last slide, so kappa casein, it does have a phosphate group on it?
One. So one, with one phosphosyl group and up to 3 carbohydrates. So it's mostly glycosylated. It's definitely not as phosphorylated as the others, so it's not sensitive to calcium.
Pam, I also had a question. From the slide, beta casein is not the least negative charge, or is it kappa casein?
So kappa casein, it has similar charge, the net negative charge basically, and they are both very amphiphilic, but this part, the glycosylation, makes this more hydrophilic than this part in beta casein, and that's why this part plays an important role in stabilizing the micelle. Okay. So they don't exist as individual proteins. So the structure is quaternary structure. So they associate as micelles. So these caseins associate together as micelles. So the micelle itself, it's made up mostly of protein. You have some salts, obviously, calcium phosphates with the protein micelle. And then the ratio, if you see here, you have mostly alpha S1 and a lot of also beta. You have alpha S2 and kappa casein at lower proportion. However, again, different variants of milk, different types of milk might have higher kappa casein content than others, for example, and the kappa casein is directly related to the size of the micelle. So the higher the kappa casein concentration, the smaller is the micelle, because it's going to be mostly on the surface of the micelle. So it's basically, you will have more micelles of smaller size, the more kappa casein you have. So that's why you have a range in diameter. And also it's porous and it's hydrated. So there's a lot of water interaction. And another thing unique about it is white. So it's light scattering.
I have a question.
Yeah.
Do you remember, or did you ever hear about that like A2 milk?
What, say that again, what milk?
A2. It was like literally called A2 milk, and they were talking about something of like, I don't remember the marketing stuff, but it was something about like the specific protein, or there was more of a protein, or they filtered it. And if you haven't heard of it, then my question might not be relevant, because I was wondering if like that's what they were referring to here, like a casein
No, I
protein, or
haven't heard
something
of it.
else.
There are genetically engineered types of milk that would have one particular type of protein versus another to remove, for potentially like allergenicity. I know. And then there is a milk that is non-dairy milk, which is the gosh, what is it? Day something. So they produce the proteins from using microbes. So they change the DNA of the microbes, and they expel the proteins that are present in milk, and they formulate non-dairy milk. I can't remember the company, but I don't know what the A2 is. Does anybody else know?
So oh, sorry about that. In the animal science department, we did talk about it. I think it's missing one of the casein. I think beta casein, but they said that it's supposed to help for some people with like either milk allergenicity or some other health benefit, but I've never actually tried it myself.
Yeah, and it is genetically engineered, because you cannot just eliminate one protein without doing anything.
Okay. Cool, thanks. Yeah, I just had that thought as we were looking at this. I was like, oh, was that the A2 they were referring to? But yeah, okay.
No.
Thanks.
Okay, so there are a lot of studies that try to determine how the casein micelle look like, or how is structured. So there are several models, but now the most believed model is the nanocluster model. But there has been different models, and I'll talk about them briefly here. So the coat core model, basically it says that in the core, you only have alpha and beta, and on the outside, you only have kappa casein. So that is the theory. And they do say, which is correct, that the relationship between the micelle surface area and the casein, kappa casein, is linear, which is still correct. But it is not correct. Apparently, afterwards, it found out that the core is not only consistent of alpha and beta. There is some kappa in the core as well. And the surface is not entirely just kappa casein. So another model came to be, and this is the submicelle model. So in here, they thought that, well, there are submicelles. So the casein micelle is made up of individual submicelles, and each submicelle has an interior core that is mostly hydrophobic, and you have some kappa casein on the exterior. But the more you have kappa casein on the exterior, these submicelles will be on the surface of the casein micelle. So you might have submicelle inside that have some kappa casein, but most of the micelles or submicelles that have the highest amount of kappa casein on the surface would be on the outer surface of the micelle itself. And in the inside, you have the calcium phosphate clusters that link those submicelles together. So on the surface of the submicelles, you have those phosphorylated groups that have the phosphate group that would link with other submicelles by calcium phosphate clusters formation. Okay, so they kind of confirmed the submicelle by looking at electron micrograph image, and they saw these little spots, and they assumed that each of these spots is a submicelle on its own. So that's how it came to be, a submicelle model. However, well, okay, this is more of the submicelle model, just showing the submicelles, those that have more kappa caseins and hydrophilic groups are on the surface, and those in the inside are hydrophobic and also interacting by the calcium phosphate clusters that link the proteins to each other and keep them stabilized together. So the nanocluster model is the one that is most believed right now today, and here there's no submicelle, no surface and core. What they say that there's the caseins are intertwined with each other, and there are kappa casein in the inside intertwined, but there's most of it is on the outside, with the glycomacropeptide giving the hydrophilicity on the outside of the casein. And these little black spots are your calcium phosphate clusters that link the proteins, and the other interactions are mostly hydrophobic interactions, with the beta casein playing an important role. So it's basically an intertwined system with all of these proteins interacting with each other, not necessarily a structured submicelle, but the same concept, with most of the kappa casein is on the outside. So this is just an illustration of the different models. So the submicelle model and the nanocluster model. So it's just a nice picture that I thought would be nice to have and see. This is a cryotransmission electron microscopy image where you can see the casein, and the dark spots are your calcium phosphate clusters. This is another image, field emission scanning microscopy as well of a casein myself. And this is a schematic drawing of the casein micelle, which will explain, which I'll use later on to explain what happens under different conditions, what happens to the proteins and how they interact or not interact under different conditions. So basically your calcium phosphate is clusters are the gray balls here, and then you have you know, the glycomacropeptide on the outside. The green spot is the other side of the kappa casein, which is the para-kappa casein. And the alphas and alpha S's and the beta in the orange are interacting with each other. And the blue part is just the hydrophobic component of beta casein that encourages hydrophobic interactions, basically.
Pam, do disulfide bridges also play a part in stabilizing the casein micelle?
No. So from the caseins, all of the casein beta and alpha S1 don't have any cysteine residues, any sulfider groups. And the alpha S2 has two sulfider groups that are just interacting disulfide within the molecule itself. So there is no disulfide interaction between the different caseins. And kappa casein also has one disulfide bond, which is important because when you heat milk and it's kappa casein is on the surface, but then if you have beta-lactoglobulin and it's denatured, you have that free sulfider groups. That free sulfider group causes disulfide interchange with kappa casein. And that's why sometimes they add beta whey protein to yogurt, for example, additional, to enhance the gelling, because then the beta-lactoglobulin react with the kappa casein. So the interesting part about casein, they're very heat stable, but once they, you go above a certain temperature, you start getting coagulation. So what happens is a lot of different things happen that leads to coagulation. And these are the different factors, but I have an illustration after this slide to show you how these different factors play a role. So when you heat, of course, you have a denatured whey protein. So the denatured whey protein is going to interact with kappa casein and form larger polymers. The pH decreases. So the pH decreases because you're forming colloidal calcium phosphate. So what's colloidal calcium phosphate? That means all the phosphate, the phosphate group has 3 negative charges. So all of them, instead of being protonated, they have calcium bound to them. And then the calcium can bind 2 phosphate groups. So you end up with a colloidal calcium phosphate. And the primary and secondary phosphates, that means the phosphates that are interacting with only one calcium, they have, the other phosphate has a hydrogen. But when you have calcium interacting instead of the hydrogen, the protons are now available, soluble, and they decrease the pH. When you have more, of course, protons, free protons, active protons, then the pH goes down. And you get dephosphorylation of the casein, which also further destabilizes the casein micelle. Lactose degrade, causing even more reduction in pH. Kappa casein can hydrolyze, which if you break the glycomacropeptide, it's also going to cause destabilization. And that will cause, when it's hydrolyzed, will cause decrease in surface charge. And therefore, you have this hydrophobicity in the micelle with less phosphorylation. The glycomacropeptide is gone. Some calcium is leached out due to the calcium imbalance because of the formation of the colloidal phosphate. Then you get coagulum, or the protein will coagulate. So with heating, this is kind of illustrating, you have a higher amount of colloidal calcium phosphate. This is going to produce more protons in the system, so this decreases the pH. Degradation of lactose also decreases the pH. Hydrolysis of phosphoserine, which is dephosphorylation, causes decrease in pH. Basically, you're getting closer and closer to the isoelectric point of the casein, which basically will lower the zeta potential. And what happens is when your colloidal calcium phosphates now are mostly on the outside of the casein micelle, so instead of the calcium being in the micelle, they're leaching out and forming colloidal calcium phosphate. They're going to be involved in interactions between micelles rather than within the micelle itself. So we get aggregation. So we get interaction via hydrophobic interaction, via calcium phosphate bridges. This is caused irreversible aggregation, and the denatured beta-lactoglobulin adds more to that aggregation by interacting with the kappa casein via disulfide linkages. Hence, the whole aggregation is irreversible. Yeah, you have strong hydrophobic interaction. So that's kind of what happens with heating. So isoelectric point of casein is 4.6. So that's where, when you form a yogurt, you kind of have your lactic acid bacteria, and it just lowers gradually the pH to 4.6, losing the charge, Zeta-Potential is lowered, casein come together and aggregate, but without fusion. That means you form a gel with minimum syneresis. That's your yogurt. However, on the other hand, you form a coagulum type of a gel, or just a coagulum really, where you don't hold a lot of water, but then you form this curd due to fusing of the casein together. So how does this happen? It's enzymatic. So again, you have your chymosin is going to break the glycomacropeptide off, and then you lose that surface charge and the stability, and then the casein micelle comes together via hydrophobic interaction and calcium phosphate bridges. Hence, the addition of calcium enhances that formation. So this is the illustration I talked about. It's going to kind of tell you what happens under different conditions. So first one here, A, is where you have everything is intact. So you have your glycomacropeptide, which causes a repulsion between the casein micelle, and the casein micelles are forming a stable colloidal system. This one here is when you have the B, is when you have, I don't know where my mouse went. Okay, here. So when you have the enzyme, so you basically cleave off that glycomacropeptide, you lose the stability, then the casein micelles come together by hydrophobic interaction and then also form calcium phosphate bridges. So the C is when you basically reduce the pH. So your glycomacropeptide loses its charge. And again, in this case, you would have attraction between the casein micelles. The last one here illustrates a denatured beta-lactoglobulin attached to the kappa casein by disulfide linkages. This is something that actually not desirable when you're trying to make cheese using chymosin. That's why they don't heat the milk at higher temperatures. So they usually heat the milk to 60 degrees rather than 70, where beta-lactoglobulin denatured at 70 degrees and higher. So what happens if you have if you heat the milk that you're going to use for cheese formation, and then you end up having this beta-lactoglobulin interacting with the kappa casein, then the kappa casein, the enzyme won't have the same, kind of blocks the enzyme, the chymosin, from being able to hydrolyze the para-kappa casein. Well, sorry, hydrolyze the glycomacropeptide to destabilize and produce the curd. So this is not desirable when you're on a farm of cheese. So you don't want this to happen. But yeah, so for enzyme produced curds, you don't want the beta-lactoglobulin to denature and react with your kappa casein. Okay, 5 more minutes and then I'll stop for the day. Okay, so storage stability. So what happens is a common thing called age gelation. So that's when you see thinning of the milk and then precipitation and formation of a gel. This is due to the endogenous, main reason is the endogenous plasmin and also bacterial proteases as well. So these would hydrolyze mostly your beta casein and cause that thinning. Another thing is during age gelation or during storage of a pasteurized milk, your beta-lactoglobulin is denatured. So during storage, that beta-lactoglobulin is gradually going to interact with the kappa casein and cause gel formation. Surface charge decreases, which enhance more casein interactions. Okay, so that's it for caseins, I guess, for now. We'll go to casein ingredients later, but we'll now talk about the structure of whey proteins. So in comparison to the caseins, whey proteins don't form stabilized quaternary structures. So they're mostly monomers, But depending on the pH, especially we'll talk about that, beta-lactoglobulin, for example, at pH 7 is a dimer, usually is interacting via hydrophobic interaction as a dimer. And at pH around the isoelectric point of weight, they form a soluble aggregate that is an octamer. So it's made up of 8 proteins, beta-lactoglobulin proteins. But that's why, so if they're still soluble, but then they will look, the water looks more cloudy than clear at around the isoelectric point. So if we look at alpha-lactalbumin and beta-lactoglobulin, so they're both very structured proteins. They have well-structured secondary structure. And then they are very globular, compact globular protein with most of the hydrophobic residues inside. Another characteristic is that the alpha-lactalbumin does not have a free sulfider group, but it does have cysteine. So it's high in cysteine residues. You have 8, and they are all engaged in disulfide interactions in the molecule. Whereas the beta-lactoglobulin has 5 cysteine residues. 4 of them are interacting here via disulfide, 1, 2 here and 1, 2 here. So this is 4. And then you have that free sulfider group, the very reactive, which is important for gel formation. So if you have alpha-lactalbumin and beta-lactoglobulin together and they both denature, this free sulfider group can cause interchange, disulfide interchange between alpha-lactalbumin and beta-lactoglobulin as well. So it's not just interactions between beta-lactoglobulin. So highly structured, minimum flexibility, unlike casein. The alpha-lactalbumin denatures at 63, and this is a characteristic of their secondary structure. So you see here there are more alpha helixes and then 1 beta sheet, whereas beta-lactoglobulin has more beta sheets and 1 alpha helix in its structure. So beta sheets are more thermally stable than alpha helixes. So there is this difference in denaturation temperature. So beta-lactoglobulin mostly denatured at an average of 73 degrees C, whereas alpha-lactalbumin denatures at 63. But definitely, as you heat, even at 60 or 65, beta-lactoglobulin start to gradually denature. But the peak denaturation is at 73 degrees. So most of its secondary structure is beta sheet, so 50%. We talked about the 5 residues of cysteine and then 1 alpha helix. Most of the hydrophobic groups are in internal moiety, but we do have 1 region on the surface, a small region on the surface that's hydrophobic. And it can bind small molecular weight compounds that are hydrophobic, such as retinol, for example. This is the reminder, you saw this before, the solubility and linking the structure to function. And because it's very hydrophilic on the surface, and when it's not denatured, regardless of the pH, even when you lose charge, you don't have enough hydrophobicity on the surface, so you don't get that much aggregation. It remains soluble. But once you denature it, once you heat it, it's going to unfold and then expose the hydrophobic group. So when you eliminate the charge at the isoelectric point, you're going to see a drop in solubility. So this is just a reminder, linking again structure to function. All right, so I'm just going to stop here today. We'll continue next time. So next week, we'll also have a group of presenters. We have Holly and Devin next week. Again, we'll start with the presentation and we'll continue with lecture after that. Okay. Do we have any questions? Okay. Well, we'll see you later. Bye.